I'm going to be discussing about DNA and its structure. So this is a DNA strand, and this is a polymer. The monomer of this DNA are going to be nucleotides. Nucleotides look like this. They have a phosphate group, which is just represented by this circle, and then they have a 5-carbon sugar, or a deoxyribose. We just draw a pentagon. So here, I'll make it connect better. And then we have its nitrogenous base, which we just draw with a hexagon. So this whole thing here, that's a nucleotide. We have our phosphate group, we have our deoxyribose, or our 5-carbon sugar, and then we have the nitrogenous base. There are 4 different kinds of nucleotides, and they're all different just because of how their nitrogenous base is. It could be having adenine as its nitrogenous base, it could be having thymine as its nitrogenous, or it could be having cytosine or guanine. So that's what makes its 4 different types. Now, there's something about adenine and guanine. There specifically looks like this. You have its phosphate group, you have the 5-carbon sugar, and then you have its nitrogenous base. But instead of it just being one ring or one hexagon, it's going to be having 2 now. So this kind of, the way it looks, is only going to be seen in adenine and guanine. These are called, this one right here, these are called the purines. The purine nitrogenous, the purine kind of model of it. And this right here, this specifically represents thymine and cytosine. This is going to be a pyrimidine. Now there is a way that they connect, okay? As you can see here, I'm specifically doing it in a way for colors of connecting. You see that the blue connects with the red or vice versa, red, blue, it doesn't matter. But there's a rule, because adenine will always connect to thymine. Adenine and thymine must always connect together. And then you have cytosine, and then you have guanine. These always connect together. It cannot be interchangeable, okay? These are strict rules. Now, yes, it's a strict rule on how they pair up. Like you see here, it's like adenine, thymine, then you see guanine, cytosine, or sometimes vice versa, right? But the way it's structured, when you line them all together and how you assemble them, that's what decides the person's traits, okay? So I have, let's see here, we have the thymine and we have the adenine, right? Next is also thymine and adenine. That's just how its traits are going to be decided. So this is it, and thank you for watching.